Hello everyone, welcome to our second biology lecture. So today we're going to be looking at the basic chemistry and the structure of nucleic acids. So we're going to be looking at how do atoms bond to form molecules, what molecules characterize our living things, what are nucleic acids, we'll look at DNA and RNA, and the functions of both DNA and RNA. So how do atoms bond to form molecules? So molecules essentially bind through different types of binding to form molecules, and these involve various types of chemical bonds. So we have our within molecules and between molecule bonds. So within molecule bonds, we have our covalent bond, which involves the sharing of electron pairs. So here's an example. We have an ionic bond, which is bonded through the attraction of opposite charges. So as you guys know, positive attracts the negative, while the same charge repels. So bonds between molecules include our hydrogen bonds, which involves the sharing of a hydrogen atom. So here we can see the sharing. Also, what's good to determine is the bond energy. So when we look at bond energy, this is the strength of the bond. So these covalent structures are usually involved in the foundation of our molecules, so these will usually not break down. So you've probably heard of denaturing of proteins, where we can break down certain proteins. This involves when we break down our weak interactions, but some are very strong and will still have that original amino acid structure. But again, just keep in mind the bonding strength. Then we have our hydrogen bonds, like I said. We have a hydrophobic interaction, which is the interaction of nonpolar substances in the presence of polar substances, especially water. Then we have our van der Waals interactions, which is the interaction of electrons of nonpolar substances. So again, bond energy is the amount of energy needed to separate two bonded or interacting atoms under physiological conditions. So the biological implications of molecular bonding. So structure. The structure of a molecule is precise. It is important to its function in cells. So usually in most cases in physiology and anatomy, structure dictates the function. So it's determined by positions of its atoms, valence orbitals. So this determines how biological molecules recognize or respond to one another with specificity. This affects its ability to react. So here we have an example where we have natural endorphin. So here we got our different molecule types. So we have our carbon, nitrogen, hydrogen, sulfur, oxygen. So essentially this example is our natural endorphin, which is released from our body. And this is how we just get our endorphin. So when we have the drug morphine, we can see here that this mimics the natural endorphin and can bind to the same receptor. So we can see based on this, we have the similar structure and also electrical charges, so it can bind into here and essentially give that endorphin response without actually being our natural endorphin. Again, that's how they use it in pain relief. So what are the function of these bonds? So the function, based on electron or proton interactions, so the function is dictated based on the electron or proton interactions. It uses gradients driven by electron and proton transfer to transfer energy, which is very important. And this is controlled/regulated by small molecules and ions, which act as signals and switches. There are different functional groups which are involved in reactions. These functional groups show more consistent, predictable chemical behavior, but functional groups show less predictable biological behavior. So here we have a bunch of different functional groups. We have our aldehydes, our ketones, carboxyl, our amino, sulfohydryl, hydroxyl, and phosphate. So we'll see some of these structures quite frequently, especially the amino acids, carboxyl groups. We see ketones in a lot of metabolism stuff. But we see, we'll talk about phosphates and amino acids and stuff later too. So how do chemical interactions play an essential role in the structure and function of living organisms? Essentially, it's quite simple that often subtle chemical changes in the types and positions of functional groups attached to large molecules can have significant biological impacts. So small changes, even at an electron and different levels like that, within a huge molecule, can make a big difference to its function. And either make it not function. That's why even small mutations in certain genes, even though they only affect one little thing, can drastically change the effect of a molecule. So we have some special molecules unique to all living organisms are the macromolecules. So as again, we talked about, there was carbohydrates, proteins, lipids, and nucleic acids. So life on Earth is based on carbon. So that's our C. And carbon is very stable. So carbon allows for the formation of large macromolecules, and the structure is stabilized by various types of chemical bonds. So macromolecules are large polymers formed by strong covalent bonds between smaller units called monomers. Complex molecular structures involve hydrogen bonds, which are H bonds. Interactions among macromolecules and ions through ionic bonds, so remember, go to that bonding page if you want a refresher, alter these molecular properties, i.e. the activity. Macromolecules are made the same way in all living organisms, which is quite interesting. So, what are the roles of these macromolecules? So, these macromolecules have a role in energy storage, structural support, transport and movement, protection and defense, regulation of metabolic activities, growth and development, hereditary, so the storage and transmission of information. The functions of these macromolecules are related to their shape and their chemical properties. So again, structure dictates function of their monomers, which are their smaller units, when they form those polymers. Higher levels of organization, i.e. we go from small to large molecules, result in the emergence of new properties. So, most macromolecules are polymers, made up of monomers, of course. So there are four major types of biological macromolecules, which are the nucleic acids, proteins, carbs, and lipids. So I would recommend memorizing that. But nucleic acid, proteins, and carbohydrates are the true polymers. Keep that in mind. So macromolecules are present in all organisms in roughly the same proportions. So you can see the proportions. So most of us are made out of largely proteins. We have quite a bit of nucleic acids, which is our DNA and stuff like that. We have our carbohydrates, mainly used for energy, and lipids in our cell membranes and other stuff like that. So reminder from our last lecture, essential features of a cell. So this is lecture 1, sorry guys. So cells can store information and transmit information. Cells have a plasma membrane, and cells can harness energy from the environment. So today we'll be focusing on this question 1. And how do they do this? They do this through the structure of nucleic acids that dictates the function. So what are nucleic acids? Well, there are two types. There's our RNA, which is ribonucleic acid, and our DNA, which is deoxyribonucleic acid. So here we have our structures. So here's our DNA, and here's our RNA. So you can see here, they highlight the difference right here. So ribose has a hydroxyl or OH group, where deoxyribose has a hydrogen, just so you know. These are distinguished by the type of sugar molecules. So these are essentially distinguished by the sugars. So, and they're also distinguished by the type of nucleotides, because RNA has uracil, or U, which we'll talk about later, and DNA has thymine, as T. They're also distinguished by their structure, where RNA is single-stranded, so you guys have probably seen the DNA. That's an RNA single-stranded, where DNA is double-stranded, coming like that. Not very good, but you can see it's double-stranded. They are similar structure in all organisms, which suggests shared ancestry. So evolutionary-wise, we probably come from the same kind of mechanisms. So what are these nucleic acids? So first, some definitions. So a nucleic acid is a base pair, a nucleoside, and a sugar, and a phosphate. But there's a difference. There's nucleosides, nucleotides, and that's mainly the difference. So the nucleotide is essentially no phosphate. So with the phosphate, we have a nucleotide. Without the phosphate, we have a nucleoside. So nucleoside, again, sugar and a base. Nucleoside monophosphate, single phosphate, diphosphate 2, triphosphate 3. So DNA. So again, DNA is deoxyribonucleic acid. It is a linear polymer of 4 subunits or nucleotides. So again, DNA is A, G, T, or C, as we can see here. These are our base pairs. So again, they're adenine, guanine, cysteine, and thymine. And these are our base pairs, which essentially we'll get to later, is they match to each other in our DNA. So they can certain letters can only match to other letters. So DNA contains a deoxyribose sugar molecule, and a phosphate molecule. So as you remember, DNA has the hydroxyl group compared instead of the hydrogen-like RNA. And we can see here, within the phosphate group, we have the ionized hydroxyl groups. So here's a structure of DNA in a different kind of picture. So here we're going 5' to 3', which we'll talk about later, but that's kind of the direction of the DNA. Then we can see the base pairs of A, G, C, and T. And these can be different for depending on different molecules. So here we have our base pairs A, G, C, and T. And you can see here our 3' hydroxyl group. And we can see that the hydroxyl group is not here when we're binding between the pairs. So just keep that in mind. So Crick and Walkins, which you may have heard about, are the ones that essentially just describe DNA structure, and we're the first ones to find it. So they describe DNA has a double-stranded helix structure with a uniform diameter with complementary base pairing. So as I mentioned, certain bases only go to, so A will only match with T and vice versa. And then it's G to C. These strands are anti-parallel, and they have a right-hand twist. So we have clues from X-ray crystallography that show DNA molecules helical and provided distance, and we have provided distance within the helix. So again, it won't be a good drawing. But again, they're anti-parallel. We have the distances between, and then here we have our base pairing. So we have, for example, so we have our A to T and etc. And then again, G to C. So here's a much better picture. So again, it's a double helix and our double-stranded molecule. There are 2 DNA strands or molecules coiled around each other in a spiral. So some of the structure, so again, we have a sugar phosphate backbone. So again, it's always when you look at DNA structures to orientate yourself with the 3' and 5', so you know where you are. So again, we can see here we have our major grooves and minor grooves. So the major groove is that bigger gap in the spiral, where the minor groove is this smaller gap. And again, here we can see our TA and G to C matching. They're color-coded here. So again, we have a complementary base pairing. So again, we can have a uniform diameter and an anti-parallel orientation. So again, these are a bunch of different structures kind of showing the same thing, but here's more zoomed in where we can see the actual binding of our, so here's the actual binding between our base pairs. We can see these different hydrogen bonds, a lot and stuff like that between them. We can see here we go from 5' to 3' and 5' to 3' as well. So here we have examples where A and T are held together by 2 hydrogen bonds, while G to C are held together by 3 hydrogen bonds. That's important to remember. So A to T, 2 H bonds, G to C, 3 H bonds. Sorry, ran out of room there. So this can be an example as a pyrimidine, and these are purines. So we can classify these base pairs. So our A to T's are considered purines. So purines, A, T, A and G, and then our pyrimidines, I'm not going to spell the whole thing out, pyrim is T and C. So again, just to clarify, pyrimidines, T and C, or thymine and cysteine, and our purines are guanine and adenine. So one rule to keep in mind is Chargaff's rule, which of complementary base pairing. So again, A will only match with T, guanine will only match with cysteine. So we see that here. So T to A, C to G. And again, 3 hydrogen bonds C to G, 2 hydrogen bonds T to A. So now moving to RNA. So RNA is ribonucleic acid and is a linear polymer of 4 nucleotides. So as we can remember, ribose has a hydroxyl group, while DNA has a hydrogen group. See there. Uracil has a hydrogen group, where thymine has a methyl group. So importantly, we can see unique to the ribose sugar, the RNA, is uracil. So instead of instead of T, RNA uses U. So here's our DNA version, deoxyribose. So in DNA, the sugar is deoxyribose. So this is a great summary figure right here. So I recommend you go through it. Where RNA has ribose, the bases are A, T, C, and G. Where instead in RNA we have A, U, C, and G. And these go together and these go together. The 5' end in DNA is a monophosphate, while in RNA it's a triphosphate. DNA is very large and RNA is smaller. Again, DNA is double-stranded, RNA is single-stranded. So what are the major functions of DNA? So DNA is an information molecule encoding heritable information. So if someone's like, oh, that's my genes, why you're so short or something like that, this is essentially where this information comes from. And this information is stored in the sequence of 4 bases of our DNA molecule, like we said, or A, D, C, G, and DNA. And we have DNA carriers, which have instructions to make more DNA through replication. The parental strands from our parents act as templates for the daughter strands. So these 2 strands from the parental strand are acting as the template strand for the daughter strand. So these daughter strands are being matched based on that base pairing. So they're being recreated. So if the parent strand has a T, it will be an A. If it's a C, it would be a G on the daughter strands. And then again, this process involves both types of nucleic acids, both DNA and RNA. But we'll get into this more detail later. So again, DNA is an information molecule. So secondly, our DNA carries instructions to make proteins through protein synthesis. So again, here's a great summary figure. So here's our parental DNA strand that we inherited. We have our transcription forming an mRNA. Then we have our translation, where we have transfer RNAs, and our ribosome. But we'll get into more detail. Again, so this information is transcribed, i.e. is copied from DNA molecules to RNA molecules. And these are translated, keyword, translated, to specify sequence of amino acids in the protein molecules. So every protein is composed of a bunch of different amino acids, polypeptide chain. So you can see that here. So it's essentially being sequenced out from the DNA. So this process involves both types of nucleic acids, DNA and RNA. As we can see here, mRNA, tRNAs, we got our DNA as well. So that's it for now. That was our brief introduction. So we'll get into more detail about this later and go into transcription and translation as well as the ribosomes. So hopefully you guys enjoyed it. See you next time.